like to thank the committee for giving me the opportunity to speak, and following two great talks from Jonathan and Annie, I'm really excited to be here, so thank you for having me. I'm going to go over brief background. I'm going to talk about our study design since it was a human study, and then kind of finish with generalizability and conclusions for these next 10 or so minutes. So I think it is important to ask these questions. We are, sitting on a virtual conference in part because of this major pandemic. Started from a SARS-CoV-2 infection in 2019. When that causes disease, that's COVID-19. But why should we as gut microbial ecologists really care about this? I think there's a number of compelling pieces of information to suggest that we should study the gut microbiome in COVID. The first is that very early, essentially this is a publication in March of 2020, shortly after the pandemic began, SARS-CoV-2 nucleic acid was robustly detectable within stool. And so the nucleic acid is there that would provide evidence to suggest that perhaps the gut microbial ecology could be changing because of the presence of that nucleic acid or active infection. As I just mentioned, COVID-19 has a subset of individuals that have enteric symptoms. How big this subset is actually an area of ongoing investigation. I think initial reports suggested that it was a substantial minority in the teen percentile, but it's probably a small minority of individuals, but they do develop, there are some people that do develop active enteritis. A third note is that the gut microbiome certainly contributes to viral immunity in the context of influenza infection. There's good human studies published now almost 2 years ago, a human vaccination study that showed that treatment with broad spectrum antibiotics abrogated influenza responses or the titer of anti-influenza responses in the vaccinated population. So, that as people that study the gut microbiome, we wouldn't necessarily think that that's shocking, but it does underscore the importance of the gut microbial community in influencing immunity. And in a final note, and probably the most important note for this study, is that in a recent study of individuals with COVID-19 from Wuhan, there was 76% of individuals reporting a symptom at 6 months after infection. Now, these were the individuals with the most severe forms of infection, the ones that were hospitalized. But that's actually remarkable. And if you think that, if you contextualize that with the fact that there's hundreds of millions of individuals with COVID that have had COVID-19, and that infection has the capacity to impart symptoms months afterwards, studying what happens after infection is critically important because this population of people continues to grow. So I hope that in this microbiome conference that this is a reasonable and compelling evidence to suggest that COVID-19 involves the gut and the host far after the initial infection, so we should study those people. This is the first of a few collaborators that were involved in this work. Solgi Lee is in the Division of Infectious Diseases at San Francisco General Hospital, which is an affiliate of the University of California, San Francisco. She helped collect a cohort of 18 individuals from whom we obtained 58 samples of stool shortly after their COVID infection. She enrolled individuals based off of a household design. And what you can see here is that some subjects were enrolled, and if they're blue, they are COVID positive. If they're yellow, they're COVID negative. So COVID positive enrolls from the household. The COVID negative individuals in that same household are given the opportunity to enroll and submit samples for stool and other types of biomarkers. We also had 7 individuals that came from their own houses where no other members of their household contributed to the study. So I refer to them as solo. As I mentioned, we collected 58 samples. We conducted 2 forms of sequencing. We did amplicon sequencing using a MiSeq Illumina machine, and then we did shotgun metagenomics using a NovaSeq instrument. One thing that we immediately observed is using permanova testing that it was obvious that there was a signal for an impact of COVID on the gut microbial community. And this is consistent with now a number of reports, primarily and almost exclusively in inpatients. Keep in mind that our patients are actually outpatients, and these samples are collected while they're dwelling within their homes. We poured over this principal coordinate analysis actually quite a bit in our subgroup meetings in the Turnbull lab. And it occurred to us that actually it wasn't just that COVID was linked to gut microbial ecology, but actually COVID individuals moved more and that they were more dispersed than individuals that did not have SARS-CoV-2 infection. This is statistically testable using the vegan package in an unbiased way. And indeed, there is more dispersion in the COVID gut microbial community. Going back to what I was just saying, if you measure the successive points of serial samples that were collected between a given individual, you see that people that had COVID actually moved a lot more between points than people that were COVID negative. And that was true regardless of the type of sequencing technology that we used. I will say for metagenomic data, it did require using data annotations that include species level indices, though species level data that it was including stratified pathways mirrored this same data, same pattern. This caused me to think about a study from Dionysius Antonopoulos that was published in 2009 in the early days of kind of the revolution of gut microbial ecology that Jonathan was mentioning during his keynote session. And this is a very important study in that in this study, animals were exposed to antibiotics and then either housed kind of with other animals exposed to antibiotics, or they were housed with animals that had no type of perturbation to their gut microbial communities. And what they saw was that animals that were housed with healthy animals, they recovered faster. And so antibiotics are a perturbation, but recovery of the gut microbiome is actually a function not just of the microbes themselves, but the behaviors of the host. Why might I think about this study? The reason I was thinking about that is because our samples were collected during a time when human behavior was measurably changing. It wasn't just that our subjects were exposed to and had SARS-CoV-2 infection, but in fact, the behaviors of the population that they were living in were very different. This is another collaborator we have here at UCSF, Yulin Swen. She helped us measure the social distancing behavior using cell phone tracking data. And so an individual or users are actually enrolled for a company, it's called Cubic. And their addresses are known and their cell phones move. And Cubic can draw a rectangle where the cell phone is and where the home is. And in that way, generate population level mobility and estimates of how many individuals are accurately staying at home. This is actually quite important because these are direct measures of population level social distancing practices, as opposed to what kind of policy mandates are occurring and actually measures of what people are doing. And what we can, and the actual movement in meter for this variable called the cubic mobility index is proportional to movement in a logarithmic scale. So 10 to the 1, or a CMI value of 1, is 10 meters of movement. 10 to the 4 is 10,000 meters of movement. So it's actually a massive difference. And what we could see here is that in our specific cohort, that we happen to be collecting samples during time periods where social distancing practices specifically for the communities from which these samples were being harvested were massively changing. This was captured in the popular media as kind of the kickoff weekend to the summer, and there was a lot of discussion about kind of we're going to get back to normal. And even though the San Francisco Bay Area generally did exceptionally well for social distancing practices, you can see here that we collected samples during a time period where people essentially started reversing those practices. Building a multiple parameter model using Permanova testing, we see that the households that samples came from, COVID status, and cubic mobility, so the level of mobility in the population, are all statistically linked to the gut microbial community. But remembering that there's actually a close association in our own data between social distancing practices and the day of the year, uniquely because we were sampling across a time period when social distancing practices were changing, we also were able to see that day position in the calendar year 2020 was linked just at an equivalent level of variation to population level social distancing practices. We would actually expect that, and importantly, it's true. This ends up being useful because it enables me to compare to other data sets where this type of information may not be present. I'll get to that in just a second. Looking at alpha diversity over time, what we saw was that in COVID negative individuals, there was actually an increase in alpha diversity over the time of sampling. But for COVID positive individuals, there was no such association. Fortunately for us, we did have other collaborators at UCSF that were phenotyping individuals as part of the immunomicrobiome cohort. They collected their samples in the 2 months prior to 2020. And what they observed, what I observed in their data, were similar associations in that alpha diversity did change over time in this group of healthy San Franciscans, and that beta diversity was linked to day position, even though we did not have social distancing data available for these individuals. So that leads me to this conclusion, which is multiple fold. One is that COVID survivors exist and they're a growing population, and so we really should be studying these people. And that individuals that were infected with SARS-CoV-2 and developed symptoms went on to have more disorganized microbial communities. And this figure is kind of helps illustrate this. You might be moving around and the healthy microbiome has natural variation over time, but with the perturbation, in this case, the SARS-CoV-2 infection, the microbiome is now perturbed. And its recovery is now compounded by social distancing practices. I'd like to acknowledge the incredible mentorship of my scientific mentor, Peter Turnbaugh, two outstanding postdocs in the lab that I've worked closely with, Cecilia Naker and Jordan Bissons, our collaborators that I've introduced you throughout the talk. I didn't mention Susan Lynch. She's been really instrumental in helping me look through my data and help me actually introduce me to various sequencing cores on our campus. Preston Tassoff did a wonderful job helping me to sequence these samples during times when not everyone was allowed to be on UCSF's campus. I'd like to acknowledge my funding sources, and finally the patients and family members